Yeah, there were actually two talks at ASH that I thought were pretty interesting from in that perspective. First, Tanya Siddiqui did update the phase I portion of the Lisocabtagene maraleucel or Liso-cel trial, phase I/II trial in CLL that has been dose escalating, and that basically showed high rates of complete remission and also very deep molecular remissions in a substantial fraction of the patients, and that those patients, especially that had the molecular remissions, have had sustained, and many of them have had sustained remissions, leading to the possibility that some of these patients could be long-term responders. Now, we have personally used the Lisocabtagene maraleucel product before it was named that, and in a slightly different manufacturing process in the work that we had done in Seattle. And we've now had people out more than 5 years in molecular remissions. And so I think that this potentially could lead to long-term cure in patients with CLL. And the UPenn group has shown the same thing from the first few patients treated with the what became the tisagenlecleucel product in patients with CLL, and they have patients out now over 10 years in complete remission. So this phase I study has now been moved into a phase II study that is currently accruing, and the hope is that this eventually will provide enough data to be able to go to the FDA. Now, Bill Weirda from MD Anderson also presented a portion of the trial that was concurrently given with Ibrutinib. Now, there have been several studies, including one of our own, that has shown that Ibrutinib may actually make a better T cell that could be then used to make a CAR T cell. And this is probably due to some of the off BTK effects of Ibrutinib. And again, the UPenn group had shown previously, and several other groups have shown that if you give patients Ibrutinib and then collect their cells and make CAR T cells, that those T cells may be more functionally fit. And so we had done a trial where we gave Ibrutinib to patients for a couple of weeks at least before we collected T cells, and then concurrently continued Ibrutinib through their CAR T cell therapy. And in our earlier phase study, we showed that that seemed to cut down on the cytokine release syndrome toxicity that patients had, and potentially lead to deeper molecular remissions in some patients. And so there was a arm of the Liso-cel trial that used concurrent Ibrutinib in exactly the same way. So patients were either restarted or continued on Ibrutinib, and then had their cells collected, and then continued on the Ibrutinib through their treatment. And again, Dr. Weirda showed very similar results to what Dr. Siddiqui had shown, with overall good remission rates, deep response rates, and we just need to follow that now longer to see which of those approaches will be better. And now at this point, they look quite similar, but I think the details will emerge as we follow these people for many years. But I think that at least in this situation, we have a product that's getting closer to a potentially commercial approval in CLL.